Okay, so welcome to this next video in which we are discussing the PI3 kinase AKT mTOR pathway. Okay, right, so let's just summarize what we've covered so far. We've seen that the ligand binding to the receptor tyrosine kinase ligand binding domain allows two receptor tyrosine kinases of the same family to dimerize together, like so. This then triggers trans-autophosphorylation, in which you get lots of these phosphotyrosine residues produced on the cytoplasmic tail. Okay, this is then going to recruit a type 1a PI3 kinase enzyme to the membrane, whilst also activating it slightly via inducing a small conformational change. This will then convert PIP2 molecules, which make up a small fraction of the phospholipids within the phospholipid bilayer, into PIP3 molecules, which are not normally present within the phospholipid bilayer. Okay, and these PIP3 molecules are now going to trigger downstream pathways. Okay, right. So they are going to recruit certain enzymes to the underside of the phospholipid bilayer. Now, the first one of these enzymes I want to discuss is an enzyme that's normally present within the cytoplasm. Okay, so it's normally floating around in the cytoplasm, and it's called phosphoinositide dependent kinase 1, or for short PDK1. So this represents phosphoinositide, that's the P, okay, and then it's dependent kinase 1, so D is for dependent, and then the K is for kinase, and then of course 1. So phosphoinositide dependent kinase 1. And I'll colour in phosphoinositide dependent kinase 1 in red here. Okay, right. Now, this is a serine threonine kinase enzyme, so let me just discuss what a serine threonine kinase enzyme is. Okay, so we've discussed tyrosine kinase enzymes, which are enzymes which catalyze the phosphorylation of tyrosine residues within proteins. Serine threonine kinase enzymes are enzymes which catalyze the phosphorylation of serine and threonine residues within proteins. So let me just remind you of the structure of serine and threonine residues. So over here, let's draw the structure of a serine residue. So once again, I'll draw the core amino acid structure. So here's the amino group, okay, followed by the alpha carbon, which will have a hydrogen coming off it, and then we'll have the carboxylic acid group down here. And the carboxylic acid group will then be linked to the amino group of the next amino acid along. Now the R group of a serine residue then has a methylene group coming off it, okay, which then has an alcohol group coming off. Okay, so this is a serine residue. Okay, and I'll underline this in purple. Right, now threonine is very, very similar, so let's draw threonine here. So here's the amino group once again, which will be bound to the carboxylic acid group of the amino acid before it. Then we have the carboxylic acid group down here, which will be bound to the amino group of the next amino acid along. And then the R group of threonine consists of a carbon, which then has an alcohol group coming off it. And this carbon will also have a methyl group coming off it, and then a hydrogen coming off it. Okay, so this then is the structure of threonine, and you can see that it's very similar in structure to serine. Okay, it's just got an additional methyl group coming off it. Right. Now, you can get the alcohol group on these serine and threonine residues phosphorylated, just like we had the alcohol group on tyrosine residues phosphorylated. Okay, so let's bring in our phosphate group. So our phosphorus atom double bound to an oxygen with two alcohol groups coming off either side, and then a single bond to an oxygen, which has then acquired an electron via ionic means and therefore has a negative charge here. Okay, right, and we're going to attach this phosphate group onto the serine residue via a phosphoester link. So we're going to, once again, take the alcohol group off the phosphate group, take the hydrogen off the alcohol group, and then bind the oxygen to the phosphorus atom. And this sort of a link here that we've got is a phosphoester link. So we're going to attach phosphate groups onto serine residues and threonine residues, and it's completely analogous for threonine residues. Okay, and this is what serine threonine kinase enzymes catalyze. Now, once again, they do not get the phosphate group that they're going to add onto serine and threonine residues from an inorganic phosphate group in the cytoplasm. Instead, these enzymes take the phosphate group off an adenosine triphosphate molecule, reducing it down to adenosine diphosphate, and then they attach that onto the alcohol group of either the serine or the threonine residue. Okay, so they don't usually get it from a free phosphate group in the cytoplasm. Okay, so that's what a serine threonine kinase enzyme does. It phosphorylates serine and threonine residues within proteins. Okay, so phosphoinositide-dependent kinase 1 is pretty active before it actually gets recruited to the underside of the phospholipid bilayer, and we're going to see that actually the reason that this now, once it's been recruited to the underside of the phospholipid bilayer, can interact with its substrate is actually going to be due to a change in the conformation of the substrate, but we'll come back to that in a moment. For now, let's just have a look at its recruitment to the underside of the phospholipid bilayer. So basically, phosphoinositide-dependent kinase 1 enzymes have a special domain, okay, which I'll colour in here. So this special domain is going to be called a pH domain, which stands for plextrin homology domain. Okay, so this is a pH, which stands for plextrin homology domain. Okay, so plextrin is a protein which is found in platelets, and we found this domain within this protein, plextrin, which you find in many different proteins, and this domain has since been called the plextrin homology domain. So it's just a conserved structure that you find in all sorts of different proteins, okay, that was first found in plextrin. Now, what's the significance of this plextrin homology domain that's in turquoise here? Well, this portion of the phosphoinositide-dependent kinase 1, and I should stress that I have just extended my picture of phosphoinositide-dependent kinase 1. This is not a separate protein. This is part of phosphoinositide-dependent kinase 1. Okay, this plextrin homology domain is now going to bind to PI3K molecules that are in the inner leaflet of the phospholipid bilayer. So let's show this here. So here's our phosphatidic acid molecule. Here's our inositol ring. Here are our phosphate groups coming off the third, the fourth, and the fifth carbons of the inositol ring. Okay, so I'll colour these phosphate groups in purple here. Okay, here is the inositol ring in blue. Okay. Here in green is glycerol, and here in orange are the two long-chain carboxylic acids which anchor it into the inner leaflet of the phospholipid bilayer. So here it is sitting within the membrane. Right, so the enzyme goes from being just floating around in the cytoplasm to being attached to this PI3K molecule that's within the inner leaflet of the phospholipid bilayer. Okay, so it's recruited up there, but binding to the PI3K doesn't really change its function that much. It still remains at the same level of activity that it was when it was running around in the cytoplasm. Okay, so we'll come back to phosphoinositide-dependent kinase 1 in a moment. A similar thing is going to happen to another enzyme, okay, and this other enzyme is known as AKT. And it also has another name. It's also more commonly known as protein kinase B. Well, I say more commonly, protein kinase B is kind of the colloquial name for AKT. If you read the research literature, actually everyone calls it AKT rather than protein kinase B. So I would say AKT is actually potentially the more common name than protein kinase B. But when people are talking about it, they generally call it protein kinase B, or they abbreviated to PKB. Okay, right. So, protein kinase B is another enzyme, which again is a serine threonine kinase enzyme. It phosphorylates serine and threonine residues within proteins. Okay, and again it starts off in the cytoplasm and is then going to be recruited to the underside of the phospholipid bilayer by the presence of PI3K molecules in the phospholipid bilayer. Okay, now, there are 3 genes which code for protein kinase B enzymes, and they're a single protein. They are a single polypeptide. They're not a complex of proteins. Okay, so the 3 genes then that code for protein kinase B enzymes are known as AKT1, AKT2, and then AKT3. And these are all separate genes. They will all have different sequences of organic bases and therefore will code for proteins which have different sequences of amino acids. Okay, they also have other names. They're also known in terms of protein kinase B, and AKT1 is called protein kinase B alpha, AKT2 is called protein kinase B beta, and AKT3 is called protein kinase B gamma. Okay, now, protein kinase B gamma, or AKT3, this gene has multiple splice variants, whereas the other 2 just have one known splice variant. So let me just talk to you about the concept of different splice variants. Okay, so let's let these 2 parallel lines here represent the 2 complementary strands of a piece of double-stranded DNA. Okay, so this is double-stranded DNA here. Now, let's say that this specific region of the double-stranded DNA that I'm boxing here, let's say that this is a gene. Okay, so I will even colour this box in, so I'll have this in blue here. Okay, right. Now, one of the strands of the gene will be the strand which is actually going to be used by the RNA polymerase II enzyme to make RNA. Okay, so the strand that the RNA polymerase enzyme actually uses to make the piece of mRNA which is complementary to the DNA is known as the coding strand. So this one that I've highlighted in purple, this will be the coding strand of the DNA. Okay, and the enzyme RNA polymerase II, which is often abbreviated to RNAP2, like so, will come in here, it will open up the DNA, it will work its way along the coding strand and produce a piece of mRNA that is complementary to that coding strand, which is in green here. Okay, so let's take this piece of mRNA out now. Here it is. Okay, now this piece of mRNA is not actually ready to be translated yet. You cannot just stick this through a ribosome and expect the outcome to be good. Okay, this is what's known as a piece of pre-mRNA, or it's also sometimes called the primary transcript because it's the first copy of the DNA. It's the primary copy of the DNA. It literally is just complementary to the DNA. Okay, right. Now the reason that you cannot just translate the primary transcript into a protein is that there are portions of the pre-mRNA that are not actually supposed to be translated. Okay, so the portions of the pre-mRNA that are actually supposed to be translated, these are known as the exons. So I'm going to box these here. Okay, so I'll get my pen out. These portions that I'm now boxing in blue here, these are going to be the portions of the pre-mRNA that we actually want to translate. Okay, and these 3 portions are therefore called the exons. Now, the portions that are in between the exons, so these 4 portions that we've got here, 1, 2, 3, 4, these portions are called the introns. Now these are not actually supposed to be translated. So if we want to have a piece of mRNA that can actually be put through a ribosome and turned into a protein, what we firstly need to do is cut out the introns and then stitch the exons back together. And this process of cutting out the introns and stitching the exons back together is known as splicing, and it will produce a smaller piece of mRNA. Okay, shown here, which will just consist of the exons put back together. Okay, so this final piece of mRNA that's actually now ready to be translated, this is called a mature mRNA. Now, in the case of some pre-mRNAs, there are multiple ways that you can splice the pre-mRNA. Okay, so when you are confronted with a pre-mRNA and you're trying to turn it into something that can actually be translated, there is multiple ways that you can proceed in the case of some pre-mRNAs. Okay, so to give a a simple example of this. Let's imagine that this final exon here is not actually essential, so you don't have to include this in the mature mRNA. Okay, so let's say that this is optional. You can either include it, in which case you'll get this mature mRNA here, or you don't have to include it. And if you don't have to include it, if you don't include it, you'll just include these two, so you'll get a shorter piece of mature mRNA. Okay, so that means that when you are confronted with this piece of pre-mRNA, you have a choice of which piece of mature mRNA you want to make. You can make the full piece with the 3 exons all sewed together, or you can make a shorter piece which just contains the first 2 exons. Now, these will then go on to code for different proteins, and these different proteins are known as the different splice variants of the gene. Okay, so this is how from one gene you can produce different proteins, basically, because there are different ways of splicing up the pre-mRNA, which will result in different mature mRNAs, which will then code for slightly different proteins. So the 2 proteins here, this one will give a big protein, and this one will give a smaller protein, basically. Okay, so most of these protein kinase B genes only have one known splice variant. Okay, so protein kinase B alpha and protein kinase B beta, those only have one known splice variant, and that protein will be called the protein kinase B alpha or the protein kinase B beta. But in the case of the protein kinase B gamma, there are 2 known splice variants. Now, the full-length one, which includes absolutely all of the exons, is called protein kinase B gamma, okay? And the shorter one, which doesn't include all of the exons and is shorter, okay? It's identical to protein kinase B gamma, but it doesn't quite have the finishing part, okay? So it's shorter. It's called protein kinase B gamma 1, and I'm sorry about having to draw it over that arrow. Okay, so there are these 2 separate splice variants then of the protein kinase B gamma gene, or the AKT3 gene. Okay, so that overall gives us 3 different types of protein kinase B enzyme. Okay, so let me now draw a picture of what the protein kinase B enzymes look like. So here is the amino terminus of the polypeptide. Then the first important domain that you have at the amino terminal end of the polypeptide is another one of these plextrin homology domains. Okay, and this is going to be responsible for recruiting it to the membrane when PI3K becomes present in the membrane. Then, after the plextrin homology domain, you have the catalytic domain. Okay, so this is the portion that is actually going to catalyze the addition of phosphate groups onto serine and threonine residues within proteins. Okay, so this is the catalytic domain here in blue. Okay, and then after the catalytic domain, you then have the regulatory domain. Okay, so we'll have this here. Now, this is the case for all of these 4 different types of protein kinase B that we've talked about. Okay, they all have these basic structures of plextrin homology domain, followed by catalytic domain, followed by regulatory domain. Okay, now, this is sitting within the cytoplasm, okay, at the moment. Now, when PIP3 arrives in the membrane, that is going to result in the protein kinase B enzyme being moved up to the underside of the phospholipid bilayer, because its plextrin homology domain will bind to the PIP3. Okay, however, that is not enough to activate the protein kinase B. To activate the protein kinase B, it needs to have certain residues phosphorylated. So there is a threonine residue within the catalytic domain, okay, and the single letter amino acid code for threonine is T, okay. And in order to be activated, the catalytic domain needs this threonine residue phosphorylated. Also, in most of the protein kinase B enzymes, you also have a serine residue within the regulatory domain, which also needs to be phosphorylated in order for the protein kinase B enzyme to be activated. So, in protein kinase B alpha, protein kinase B beta, and the full-length protein kinase B gamma, you have both a threonine residue and a serine residue, which have to be phosphorylated in order for the protein kinase B enzyme to be activated. In the case of protein kinase B gamma 1, I told you that protein kinase B gamma 1 is a shorter version of protein kinase B gamma. It's actually so short that it doesn't have this serine in the regulatory domain that needs to be phosphorylated, so it ends somewhere like this, okay, halfway through, and then doesn't have that serine residue. So all it needs is this threonine phosphorylated. Okay, right, so let's now talk about the activation then of protein kinase B. So it does begin with the protein kinase B enzyme being recruited to the cell membrane by the plextrin homology domain, which will bind to the PI3K molecules. So let's draw this. So we'll start by drawing our PI3K molecules. So here are our long-chain carboxylic acids. Here's our glycerol molecule, our phosphate group, our inositol ring here, and then the 3 phosphate groups which come off the 3rd, the 4th, and the 5th carbons of the inositol ring. Okay, so let's colour these in. Here are the long-chain carboxylic acids in orange. Here is the glycerol molecule in green. Here are the phosphate groups in pink. Okay, and here is the inositol ring in blue. Okay, right, and this is sitting within the cell membrane here. Okay, now what's going to happen is the plextrin homology domain is going to bind to the PI3K molecule here. So this is our pH domain, and I'll just label it the PIP3 here as well. Okay, and then we've got the amino terminus of our AKT enzyme here. Okay, then we've got our catalytic domain, which I'll show here. Okay, and then we've got our regulatory domain. So we've now recruited our protein kinase B enzyme to the underside of the phospholipid bilayer. Now it's going to be very close by the phosphoinositide dependent kinase 1 enzyme. So let's colour these portions in. Here is the catalytic domain in orange here. Here's the regulatory domain in green here. And here's our plextrin homology domain in turquoise here. Okay, so what now happens is the phosphoinositide dependent kinase 1 here that's been recruited to the underside of the phospholipid bilayer is going to phosphorylate the threonine that is on the catalytic domain. Now, the reason that the phosphoinositide dependent kinase 1 was not able to phosphorylate the threonine residue on the protein kinase B when it was in the cytoplasm, so remember before PIP3 arrived, both of these enzymes were within the cytoplasm. Okay, how come phosphoinositide-dependent kinase 1 doesn't phosphorylate the threonine residue when it's in the cytoplasm? Okay, well the reason is not that the phosphoinositide-dependent kinase 1 has to be activated by binding to PIP3. It's active in the cytoplasm. What actually changes is the protein kinase B enzyme. Okay, so our AKT enzyme, or protein kinase B enzyme, is going to undergo a conformational change when it binds to the PI3K, and this is now going to make this threonine residue available for phosphorylation by the phosphoinositide-dependent kinase 1. So it's subtler than you might imagine. It's not that that phosphoinositide-dependent kinase 1 undergoes a conformational change when it binds to the PIP3, which activates it. It is instead that the substrate, the AKT enzyme, undergoes a conformational change when it binds to PIP3, and then that becomes a target then for the already active phosphoinositide-dependent kinase 1. So PDK1 is going to stick a phosphate group onto the threonine within the catalytic domain of the AKT enzyme. Okay, now, I told you that in the case of most of the protein kinase B enzymes, that is not enough. Okay, so in the case of protein kinase B alpha, protein kinase B beta, and protein kinase B gamma, that's not enough. You also need the phosphorylation of the serine. Now, this is not carried out by the phosphoinositide-dependent kinase 1 enzyme, and it's still extremely controversial as to exactly how the serine within the regulatory domain of protein kinase B enzymes actually gets phosphorylated. Okay, what we do know is that it does get phosphorylated, but we don't fully understand how it gets phosphorylated and how that relates to the whole pathway. But it does get phosphorylated by something other than phosphoinositide-dependent kinase 1. Okay, right. And now what happens is once the protein kinase B enzyme has been phosphorylated on both the threonine in the catalytic domain and the serine within the regulatory domain, that causes the whole thing to now detach from the underside of the phospholipid bilayer. Okay, so it only binds there temporarily, and now it's going to detach and go off back into the cytoplasm, basically. And in the cytoplasm, it's now going to activate its target, which is the mammalian target of rapamycin complex 1, which we'll begin discussing in the next video.